317, and we're going to be looking at chapter in Pearson's book for you folks in section 58 to 157. So we're on page 68. You have a Word document, and that's the contents that I want you to know about. Just want to go over some things and maybe say it a little differently. And some of you have indicated it, well, that helped. It opened up your understanding. So we're going to try that. As you look on page 68, up there at the top where it says the cell, you come down to that first part where it says our cells are the smallest units that can carry out the function of life. It's amazing what goes on inside a cell. Just, it's mind-boggling. That's one of the reasons I enjoy this course. I just marvel at what a cell can do. And they'll tell you that you've got trillions of cells that make up your body. The interesting thing about that is there are 10 times more bacteria, fungi, and viruses that exist either on our body or in our body in some respects. They don't get in the bloodstreams as far as we know, but they can be in your respiratory tract, they can be in your ears, down your GI tract, and so forth. And so we are I guess you could say maybe a walking incubator for those, and we've got all those organisms. So, these are researchers who are looking to see what kind of organisms are growing. Get you sort of focused on fill in the command here, identify the three main parts of the cell. So you're looking for division in the textbook that we talk about, three main parts. There are a lot of parts to the cell, but they talk about the three main parts. But you don't have the other parts, you're dead in the water. So anyway, you can check off number 1 and number 4. We'll get into some of those organelles. But you come down underneath those learning outcomes into that paragraph. And it talked about how structure and function go hand in hand. You change the structure, then the processes of cells. And you want those. Catalyzed goes and catabolism. So you want to remember those. Those are back in the beginning. And also, you look over on page 69 and you see the transport of substances. Cells make substances that we need, might be hormones or something like that, might be proteins, and they need to transport those from one place to another, out of the cell, maybe into the extracellular fluid, that'd be the fluid outside the cell, into the bloodstream or whatever. And so we'll see they also transport cell to cell. We'll see that in a little bit. It's just fascinating. I'm glad I like this. It's great getting paid for what you enjoy doing. And so communication on page 69, cells literally communicate with each other. They're even finding out that bacteria communicate with each other chemically. That's their voice. And they have receptors on their surfaces that pick up chemical messages. It just blows your mind, because when you think about communication, like I'm communicating to you and you communicate to me through your emails or through taking of the tests, we think of intelligence, don't we? And you have to take a step in that direction, say bacteria, they're intelligent, and our cells communicate with other cells. Don't think science has got it figured out. a little bit, and we can help people to reproduce, and that seems to be the end of it. They don't know why. Read the first sentences. They talk about the three main parts of the cell. There are a lot of parts. Basic component, plasma, and you see the nucleus. So if you see something about what are the three basic components of a cell, plasma membrane, nucleus, and the cytoplasm. So as you look at plasma membrane, got a number of functions. And as you read through there, it talks about support, communication with other cells, and even cell here separating the inside of the cell from the outside of the cell. So it's like a fence, some sort of a barrier. And so through that barrier, that plasma membrane, certain things can come in, certain things can go out. Certain things can't come in, certain things can't go out unless they have some help. So that's basically what we want to say about that plasma membrane right now, except you got to call interstitial, that's between tissues, interstitial fluid. And so we have a balance. Plasma membrane is involved in that. Come on down to cytoplasm, and the cytoplasm. Dehydrated me, I weigh about 215 pounds. Then you take 80% of that, there'd be about 40 pounds left. That's what I would weigh if all the water was taken out, somewhere around 35. All kinds of things like proteins. We've got, as you come down on the same, on page 69, you want to come down to about 1, 2, 3, 4 lines, 5 lines. It talks about the cytofunctions, and including storage. We'll talk about storage a little bit later. Providing a site for critical reactions, including protein synthesis. All kinds of synthesis reactions, hydrolysis reactions. That all goes on in the cytoplasm. And you come on down a little later to the next paragraph, and you see it cytoplasm. We're not going to get into that really. And you see it talks about enzyme catalyzed reagents, glycolytic catabolism, lytic you see in there. So we're going to break glucose up into all kinds of components and then use it for our own body. And you see it says glycolytic catabolism, glycolysis, into smaller units, into those monomers that we can turn into energy for us. You come on down some more. In that same column on page 69, second column, you see the bold print term organelles. And I like what they say, they're molecular machines. They are molecular machines, and they carry out particular functions. So in a sense, we really have a team of organelles inside the cell, and they each have their functions and so forth. And so they all play their role. They work together under organelles. Come down on the counting on the right, 1, 2, 3, 4, 5, 6, 7, 8, and you see it says compartmentalization is also critical for survival. Now we don't have rooms in the sense of compartments, but it says Many of the organelles contain enzymes and chemicals that could damage the cell. But we have those chemicals surrounded, believe it or not, by a plasma membrane. And it contains those substances so that it doesn't damage the cell. But you'll see a little bit later that some of those will say compartments, but they're not like rooms in here with walls. But they'll attach to each other and they'll turn loose some of those chemicals and will digest something that the cell has ingested or eaten. So it's a means of protection against some. It's good for us, and yet used in the right way, activities get accomplished, and we end up having plenty of energy and so forth and all the substances that we need. I don't know if you've ever watched the movies, not movies, but the it's sort of like a documentary called Modern Marvels, or How's That Made? Those are really fascinating shows because they, as I said, I think in the Word document I sent to you, somebody had a vision and they created it in their mind, and then they got people who could metal work and plastic work and knew about lubricants and things like that, and they set up a whole business in that building. Same thing happens inside our cells, and we don't have to think about it. They are programmed. The cells have some protein filaments that help them stay in a particular shape, but they also serve in other purposes. They'd be a little different in terms of, say, the shape proteins from the transporting proteins. As a matter of fact, look at the last sentence on page 69. The protein filaments of the cytoskeleton also function as the cell's molecular highway. How many of you have ever ridden a chairlift? Maybe you went skiing up in North Carolina, something like that. We have protein filaments where substances are transported along those filaments from one part of the cell to the next. It goes to the right place. And when it gets there, I don't want to say someone greets it, but that substance gets metabolized in some way that is going to be beneficial for us. It's a wonder inside that cell. So we've got the plasma membrane, we got the cytoplasm, and all those things. That's a whole story in itself. You can go take a course called cytology, and I remember taking something like that back when I was an undergraduate at school. I was speechless. So much to learn, and we've learned even more. That was 40 years ago. So anyway, look on the next page, and you see their nucleus. And as it says, the nucleus, notice in the second line of the third line, it's actually the second line, go to the right, it says the nucleus is surrounded by a phospholipid bilayer. You're going to see more and more of these objects, these components inside the cell, and they all have the same plasma membrane, a phospholipid bilayer. Now here we are talking about phospholipids that you had to learn in chapter 2. Go back and review that. Somebody might ask you a question or two about those things, but you're building on what you learned in 2. If you haven't got it down in 2, then there'll be a tendency to be a little bit confused maybe for you. So go back and review it. Test 2 will have some of test 1's content in it. We'll weave it in there. But you notice it says they call it a nuclear envelope. It's really a plasma membrane. It's a little different. You'll see it in a few, not today necessarily. But you see it says the nucleus contains the cell's deoxyribonucleic acid, which we talked about in chapter 2. And that deoxyribonucleic acid is, I should say contains, maybe I should say is, it's the genetic material that has the blueprint for our body. Just like if any of you have ever had the enjoyment or maybe the struggle of building a house, somebody had to have a blueprint, and they had to follow that blueprint so that you got what you wanted in terms of a house. And your house looks different from somebody else's on the block because you had a different blueprint. The same thing is true here for us, but there are a lot of similarities, right? We all have noses up here. They don't show up under your armpit, do they? No, we all have a similar body pattern. It's amazing stuff, amazing stuff. So you got DNA in there, and you got RNA, ribonucleic acid, which we talked about back in chapter 2. So when you see there underneath there, the quick check, what are the 3 components make up most animal cells and human cells? And what are their functions? Think about that. See if you can tell the closet door. What's the function of the nucleus? What's the function of the cytoplasm? What's the function of the plasma membrane? Then they have this brief little blurb here coming down underneath the quick check. You see cell diversity and size. And some cells can be pretty long. Others are very short. But if you look over to the right, up the top in the second column, you see 4 examples of different cells that are present in our bodies. Notice how they're shaped, and you can look at them. They're different and are diffusion through from one part of the body, through to the other part of the body, epithelial cells. But you get the picture there. A lot of diversity. Liver cells would look different from that. Spleen cells might look somewhat different. So anyway, on page 70, and you see down at the bottom it says module 3.2, structure of the plasma membrane. Now you probably got a little dose of that when you were in Bio 110. If you took Bio 110, or if you've had a Bio 101 class, that certainly would help you. So what you want to do is you look at those learning outcomes. We're not going to get too deep into this, but you do want to know certain things. How lipids are distributed in the cell membrane, what is their function, and how carbohydrates and proteins are distributed in the cell membrane, and explain their function. So as you look over, and then of course on top of page 71 it has number 3, the overall structure of the plasma membrane according to the fluid mosaic model. So you come down to the phospholipid bilayer. That's what our cell membrane is. That's email, so don't worry about it. I'll get to it in a few minutes. Flashback. You just had an exam on these things. Go back and you want to look back over. And by the way, if you want to look at some of your tests, I've got a way to let you look at the test and see what you missed so that you correct your thinking on that, so that you don't have the same problem with the same question or similar question next time. So just shoot me an email if you would like to look. And I've got a series of steps that I've got to go through, and I try to help you look at what you missed. But you recognize these terms. Hydrophilic, hydrophobic, amphiphilic. What's a molecule that's amphiphilic? Oh, you should be thinking about that. We're getting into this. What's a phospholipid? Remember that back in chapter 2? Okay. So I want you to read this paragraph under phospholipid bilayer and come over to the right, And you see it ends over here with phospholipid bilayer. But look at the pictures at the bottom. Look at those images, figure 3.3. And you probably recognize the phospholipid molecule, got the little round polar head, and then the little tails that come off that are fatty acids. Remember, those are non-polar. Excuse me. And what they've done is they've made an illustration, produce in part B, a monolayer forms when phospholipids are added to water. So if you had a test tube and you put phospholipids on top, they're not going to mix, right? You can't get polar molecules to dissolve in nonpolar, and you can't get nonpolar to dissolve in polar. Remember, like dissolves like. And so they give you a little magnified version there in that circle. And you see it says the polar heads face the water. Remember, those are the ones that have the, well, what does it mean? You tell me what it means. Think about that. So you want to know those definitions. We use those words. What's polar mean? Oh, you want to get it. Okay. I'm not going to tell you that. So you see the polar heads face the water, and then the lipids, the nonpolar tails, face the air. So now, in the next picture, they put some more water. They didn't just pour it in and shake it up like that. They drop some water on top of it, and look at the magnified area. You see it says phospholipid rearrange. And so you see that when you put water on top of them, that phospholipid layer doubles itself. Phos swing up, and the tails meet each other, and the little blue balls up there are the polar molecules. Now, having stopped, when you look at the quick check, it asks you a question. How do the phospholipids arrange themselves in the plasma membrane? Why? Think about what we just said. Read through that section in just a couple of sentences. That's all you need. A couple of sentences. Be okay. Now, as you come down to the fluid mosaic model of the plasma membrane, it says, what is cholesterol? Well, cholesterol is going to be part of this membrane. Remember, excuse me, everybody thinks, oh, cholesterol is horrible. Some people don't care about it at all. But others think it's, oh, it's horrible. I want to get rid of it. No, you don't want to get rid of it. You need it. We need it. Okay. But what is it? Chapter 2. What are glycoproteins and glycolipids? Can you figure that out at this point? We've never really talked about those. But think of the two words that are put together to give you that one word, like glycolipid, glycoprotein. Think about it. Try to figure it out. Not hard. Now, come down about 3 lines, and you see it says scattered through the phospholipid bilayer. Now, look at the picture below. You're looking at figure 3.4. You want to be able to look at that and identify components of this view of the phospholipid bilayer. Come down to number 3, line 3. Scattered throughout the phospholipid bilayer of the plasma membrane are various proteins, other kinds of lipids, and carbohydrates. All these components together are thought to resemble a mosaic. Now, I think in the Word document, I think I mentioned how people take different colored beans. Some are black, some are white, some are red, some are brown, and so forth. And they arrange them in a pattern, which you would call a mosaic. Some people take stones that are colored differently, maybe painted. And so they're tiny little stones maybe, and they put them in a particular pattern, glue them on a board, and someone says, oh, I like that, I'll give you $50 for it. It's a form of art. They use the little stones to make some sort of a presentation. Might be a little house on a board. Now, I'm not talking about a three-dimensional type thing, but stones just glued to a board so that you see a pattern. Could be a rooster, could be a horse, could be a barn, could be a tree. It's whatever they create in their head. But that's a mosaic. So when people have looked at the phospholipid bilayer, they've thought of it as a mosaic. As you look at these little, come down to that picture, figure 3.4, and you see the little round balls, the little blue round balls, got some in the middle. Those phospholipid molecules are not stationary. They're all the time moving around. It's a dynamic membrane, fluid, okay? Fluid, they move very easily. They're not locked into a position like bricks on a house. They are, let's see if I can get that to do. There we go. That's a little better. They float next to each other. Of course, if you dry them out, they're not going to float. But they tend to move amongst themselves. Nothing wrong with that. That's just the way it is. And so they say fluid. So these phospholipid molecules are, and on the phospholipid bilayer. So you see these purple structures. They stand out pretty good against the yellow of the lipid portion, don't they? But you can see they're either called peripheral proteins or integral proteins. Peripheral means they stay on the surface. Integral means that they penetrate the membrane. They go all the way through from the polar part on the outside to the polar part on the inside. Okay? Those proteins go all the way through. And so you see those are called integral, that go all the way through. If they just stay on the surface, they're called peripheral. When we talk about the periphery, that means the edges. Not in the middle, but in the edges. Okay? On the layer, outer and inner layers. And then you see these little things that look like little hexoses, glucose, the galactose, and things like that. See those little green things? Carbohydrate chains. Those are like glucose molecules or galactose molecules hooked into M. So as you look, you see some proteins that are hooked to the carbohydrate. Glycoprotein. Glyco, like glycogen, okay? That's a big molecule with a lot of glucose molecules attached to it. And then you see glycolipids over on the right. That's a sugar combined with a lipid. So it's got it's called a glycolipid. It has its purpose. And you see these transmembrane proteins. Those are integral also. And they play a role. Let's look. Now we know that the purpose of the plasma membrane is to control what goes in and out. Over on page 73. And you see a number of pictures up top. It's figure 3.5, functions of membrane proteins. Okay? Look at the top one. transporting substances into or out of the cells. Go to the right. Some act as receptors, and they pick up a molecule and they put it inside. And then E, those are proteins that link cells together. What's the difference between 5 examples that we see in terms of their function? Well, you'd say, okay, Mr. Pritchett, this looks like this one kind of helps things get through the cell membrane. What's the difference between those proteins structurally and chemically? You had that on the last test. It really matters that you use the right amino acids in the right sequence and they have the right number. You change that, you change the shape of the protein and you change its function. We can make thousands of different kinds of proteins. And we need it too. You can see those functions down there at the bottom of page 73. It's just amazing stuff. See, look at those linker proteins. That helps them stick together. When we get into the integumentary system, we'll look at some of those in a little bit of more detail. Okay, so let's go to page 74. We're getting close to the end here. I'm only going to go for about 45 minutes. So that first column, and you see the word cholesterol, italics, but you see cholesterol, and notice its function. It has a role in our body. What kind of substance is cholesterol? Is it a phospholipid? Is it a protein? Is it a carbohydrate? What is it? You remember that 4-carbon structure? It's coming back. Don't forget it. That's why I say go back and read the old material. Oh, I got to get it straight in my head. And you try and connect what you're hearing here, what you're reading here, and we'll be reading for this week. So you keep that picture, keep it fresh. You're going to be using it all semester. Come down into the second column, not the second, excuse me, the first column, come down the second paragraph. And you see in that little paragraph, it has in italics, glycolipids, glycoproteins, and so forth. I want you to come down 1, 2, 3, 4, 5, 6 lines, and you'll see this term cell recognition. Cell recognition. They identify the type of cell. White blood cells have to identify cells. That's just one example. But if you had a kidney problem and you needed a kidney, I said, well, I'll give you mine. And everybody said, okay, that'd be great. It might not work. There's a good chance it wouldn't work because my proteins, my glycoproteins and so forth are different from your glycoproteins. And we couldn't perform that tissue transfer, that organ transfer. Because even though mine is a kidney, and it's great, works great, been working good for 73 years, but it's not yours. Your body would recognize my kidney as foreign, and your body, your immune system would try to destroy it. We have to match up to a degree those proteins. If we don't, You're just wasting my kidney and wasting your time on the operating room table. So you've got to get those things matched. We don't do it perfectly, but we do it enough so that there is a bit of a rejection, but we can control it chemically. You get into micro, we'll talk a little bit more about it, a little bit more deeply. Okay, module 3.3. We started out with 3.1 and we're already over to 3.3. And I think you probably have before in terms of, I think that Bio 110. But learning outcomes, get those up here. So you're looking for various components in this chapter. Graph on page 70F, extracellular fluid. Here's where our membrane comes in and it monitors things so that homeostasis is maintained within the cell and without the cell. As you come down about, you can read that paragraph and it'll talk about the things that are dissolved in the various fluids, our extracellular fluid. But you come down to the very last little paragraph. Substances cross the plasma membrane using several mechanisms. Now, this has all been mentioned. I'm just going over it a little bit with you. And sometimes I say it a little different from what I wrote down in the document. And you go, oh, okay. I think I got it now. So you can see it says some transport mechanisms called passive transport. They do not need energy to be expended in order for that transportation across the membrane from the outside to the inside or inside to the outside. Passive, you ever known a passive person? You just can't rile them up. And so just very laid back or whatever. Well, you don't have, in this case, it means you don't have to spend any energy. You get the micro, it'll mean that you picked up somebody else's antibodies, like they're doing with the coronavirus. They're getting people who have overcome the disease, and they take their blood, which has proteins in it, that they produce to stop the very antibodies attack the coronavirus and help that person out. Pretty neat stuff, isn't it? And so you go to the top of the next page and you see it says other mechanisms called active transport. The reason we got to eat. We have to have. I want you to read this section on diffusion. Get a definition for it and look at examples as you read through page 75. Diffusion takes place because of something we talked about back in chapter 1. The title was gradients. Gradients. You want to keep that in your mind. It's not going to go away. You got another semester and 2/3 to go, and gradients are going to be involved in all of that. So as you come on 76, notice up at the top, 76, that's first sentence, generally smaller particles in the gaseous phase, not a liquid phase, higher temperatures and steeper or greater concentration gradients increase the rate of diffusion. In other words, the more you got on this side than compared to what's on the other side of this semi-permeable membrane, that's going to make the diffusion process occur more quickly. So get a definition for diffusion, and then you come down to the next paragraph on page 76, first column. You see simple diffusion and facilitated diffusion. Mostly involves, as you see there, non-polar solutes. Water is polar, but oxygen and CO2, no, they don't have a charge on either end of them. And they go through the phospholipid bilayer without any assistance. Facilitated diffusion, you see that's going to involve charged, polar, ions, glucose, and they need a membrane protein to help them. They have a nice picture down at the bottom. Simple diffusion on the left. Nonpolar molecules go through. They're usually small. And then you see in B, facilitated diffusion of ions. Ions, of course, would have a charge on them, but you've got some proteins that help them get through. And then you see over here in the second column, I want you to look at the second column up at the top. It says in protein channels, that's what they're talking about. Now, I don't want you to know this. You just see amino acids, proteins, okay. In the protein are oriented toward the interior of the channel. Don't go memorize that. It's just the more we study it, the more we find out why something happens. But come on down to the fifth line. 1, 2, 3, 4, 5, fifth line. Most channels, which are proteins, are specific for an individual solute. So there are a number of different proteins that we have in our phospholipid bilayer, and they're designated to kick out or kick in, take out, bring in specific substances that are hydrophilic, that have a little charge on them. We don't understand. Matter of fact, if you look at the it says cell understood works. I never thought of that. You're going to be saying it a lot more in your life. I say it still now, and I'm just marveling. Okay, well, what we want to do is just mention this a little bit about osmosis, get a definition for it. And I want you to read that paragraph under osmosis. It's going to talk about water passing through the membrane. And of course, when we talk about osmosis, we're talking about basically water moving, aren't we? Yeah. We're talking about basically water moving through a semi-permeable membrane. So I want you to read about that. And I want you to read over here on page 77, down at the bottom, figure 3.8, passive transport, which is osmosis is another type of transport that doesn't require energy. But why does it move? I want you to understand, why does water move from one area to another? And then the last thing I want you to do is, as you look over here under, excuse me, column 2 on page 77, you see osmotic pressure. You want to read that, understand it. Tonicity in the last paragraph there, and you'll see isotonic solution. On page 78, a hypertonic extracellular solution. Over to the second column, a hypotonic extracellular solution. It's going to help you understand the movement of water. I think in my Word document, I asked you how many of you like syrup on your pancakes. And so this is what we're talking about. You ate those pancakes, and most of us like syrup on there. And after we eat that, we find out, I need to drink something. I'm dry in my mouth. Osmosis has taken place. Why has osmosis taken place? Think about how our parents, maybe not our parents, our great grandparents, how did they preserve meat? They didn't have refrigeration. Ask your mom and dad. See what they say. Okay, so I'm going to get another, about a 45-minute video. We'll start on page 79. So I'm going to stop here on 79, page 79. That'll get a little bit into active transport, where you have to spend energy to get something across the membrane. Okay. All right. Well, you guys have a good evening, and I'm going to try and drop this into D2L. Got the little instructions that Mr. Malachowski gave me, so hopefully I'll get that into all three of the sections. You guys have a nice afternoon. Okay.